All right, everyone. Thank you for joining us again. We are now going to move on to our drug development roundtable discussion. And this is going to be moderated by Dr. Tiongson, and our panelists are off to the right with their camera. So Dr. Tiongson, I will let you take over.
Yes. All right. So we have a panel of people from the pharmaceutical side, just because it is important to have different options in treating myasthenia, as many of the patients and families have discussed already on the chat. Having more effective ways to treat myasthenia and kind of advancing research and progress is very important. So I'd like to have we have a really great group of people here today, and I'd like to have each of you introduce yourselves. We could start, I don't know if the same video order is the same for everybody, but I have on my order, Dr. Wolf. You can go ahead and introduce yourself.
Can you hear me, first of all? I just need a head nod. Can people hear me?
Yeah.
Good.
Yeah.
Okay. I'm Gil Wolf. I'm a professor and chair of neurology at University of Buffalo, State University of New York. And most of my career has been focused on myasthenia gravis and a few other immune-mediated neuromuscular disorders. But I do cover pretty much the full spectrum of neuromuscular disease.
Okay, and next I have Frances Kipperman, who's here with us today.
Hello. Yes, my name is Frances Kipperman. I'm a patient living with MG. Briefly, the background to diagnosis took 27 years. I'm seronegative, refractory MG, so it's a bit more complicated. And I'm also very interested in the myasthenia process and how it psychologically impacts a patient and researching that.
All right, and I think Marie is having difficulty with her video, but can we hear you, Marie?
Yes, and it's working now.
Okay, great, excellent.
Okay, so my name is Marie-Hélène Jouvin. I am a physician and an immunologist, and I lead the Myasthenia Gravis program at a small pharmaceutical company called Momenta in Cambridge, Massachusetts.
Okay, great. And then next we have Ahmed Anayatullah.
Thank you. Thank you.
Yes. Sorry, I tried.
No, done very well. So good morning, good afternoon. My name is Ahmed Anayatullah. I'm a senior medical director with Alexion Clinical Development Neurology. I lead some of Alexion's programs in myasthenia gravis, including an ongoing phase 3 study. And the only focus of my group is really to develop and deliver innovative medicines for rare and devastating neurological disorders.
Okay, great. And then next, Tyler Story from UCB.
Hello, everyone. I'm a clinical researcher and neuropsychologist by training. I currently head up our medical affairs group in neurology at UCB. UCB is a biopharmaceutical company where we have a long history of working in immunological diseases and neurological diseases, and more recently have been dedicating a lot of discovery and research efforts in neuromuscular diseases like myasthenia gravis. So I'm very pleased to be here today.
Great. And then last but not least, we have Dr. Jelinas from Argenx. Oh, you have to unmute. Yeah.
I'm Dr. Deborah Jelinas. I'm a practicing neuromuscular physician. I run an ALS clinic at UNC Chapel Hill. I'm also the executive medical director for Argenx in neurologic disease. And Argenx is a new company with immunologically based therapies right now in myasthenia gravis that we're testing. Thank you.
Great. Thank you, everyone. So discussion ground rules for today, just for everybody on the call today. This is a discussion of thoughts, and we are not offering specific medical advice for anyone today. We don't plan to discuss specific products by name, but more kind of on the general drug development process from all sides. So that's why we have pharmaceutical representation, clinician representation, as well as a patient representation, just to kind of discuss these in general. So we'll get started with just some guided questions first, and then we'll go to a more broad Q&A. So just for the first question, what has the clinical trial experience like been for myasthenia? What has your experience been with the clinical trial landscape nowadays? And anyone can open up the discussion here.
Well, do you want to start, Deb, or I'll start?
Oh, no, you start.
I'll give an overview. You know, I've been involved with MG trials it was one of my first grants way back in the early 90s when I finished my fellowship out of Penn, and I was working with Rick Barron in Dallas. And we embarked on a trial of IVIG into myasthenia gravis. And I saw that there's some questions on the chat about IVIG. Anyway, which is still not approved for MG, but anyway. This is the most active time I've seen in clinical trials for myasthenia gravis. There's a lot of studies going on with various molecules with different mechanisms of action. There's a lot of excitement with FcRn blocking agents. And so it's great. And we had our first drug approved in my lifetime, and I'm old, for myasthenia gravis just 3 years ago, which is a C5 cleavage inhibitor, eculizumab. So it's great. We have a growing armamentarium. It's going to continue to grow. We're always going to have agents that are off-label as well that we've used for conventional therapy, but it's very exciting, and so it's opening up new horizons, and I'll let others pipe in.
I want to echo and just say that from a company perspective, it's been crowded because there are so many wonderful research trials going on by many, many companies. So it's an exciting, good time.
I can add to this. I think it is truly a very exciting time for both physicians, industry, and patients right now. In spite of this, I believe there are still a lot of unmet medical needs, and so there is still a lot that remains to be done, in particular for pharmaceutical company. And I think in terms of conducting clinical trials, I think there is a particular challenge with myasthenia gravis because of the fatigability that patients experience, and so you need to have a lot of sites so that patients don't have to travel too much. And then the sites have to organize themselves so that they can conduct the visit early during the day so that the patients are not tired. And then they should stick to the schedule because you want to have reproducible assessment from baseline to the visit on treatment. So there are challenges that are really important for pharma to take into account, and that make working in this field really intellectually challenging and really interesting for us.
I think I would just add that with this growth in investment and research in myasthenia gravis, it also, I think, helps raise awareness and the voice around what patients are facing and what are some of the remaining unknowns or questions that we try to address. And in addition to bringing new therapies to the market, one of the benefits of doing clinical research is that other questions are posed or answered, and then data is released. And so the discussion around this disease state ideally is elevated, and you learn more as a community, and there's more opportunity for growth and development.
Yeah, and maybe I'll, I think building on everything been said already, I think we are in a much better place today. I mean, myasthenia gravis used to be an obscure disease, and of course today it's very well characterized. we have a much better understanding of the pathophysiology. And even though, you know, we had some understanding, you know, since the 70s, it took a long time to translate that to therapies. But I think we're here. We're in the middle of it now. It's crowded space, but this is all great news for the patients, and hopefully, you know, the potential for you know, for many patients to be able to regain control of their lives in the near future. You know, if some of the ongoing efforts, you know, become realized and we reach, you know, we were able to deliver something to the patients in the near future.
Great. So I think this was already touched upon, that the challenge with developing drugs for MG in the clinical trials is the fatigability, but also from all of your perspectives, what is the challenge, the biggest challenges to develop the drugs for myasthenia?
Yeah, I can start with some of this, I guess. So, you know, in a rare disease, and MG is a rare disease. It's not ultra rare, but rare. It is challenging already to run studies in rare diseases because, you know, you have to run smaller studies. You're dealing with a smaller population, and all the challenges that come with identifying endpoints that you can use in such smaller studies, and the innovative design that we always have to, you know, seek. Everyone just said it's a crowded space, so that makes it even more challenging. So now you have more and more studies targeting, you know, the same small population. So I think that increases the challenge. I think, you know, the community is aware, and we are already working on ways to identify better endpoints and ways to enhance study design. So I think that's one thing. Maybe, of course, we have to touch on COVID-19, right? So, you know, that just, again, adds to the challenge. I mean, we've seen impact on studies being stopped or delayed, and sites, you know, suffering from restrictions to seeing patients or the type of assessments they can do. So I guess the role of remote and virtual tools here is something that we're trying to leverage, and hopefully, you know, being able to mitigate that. The last thing that I will touch upon is because it's a crowded space, and we're working on a number of modalities, a number of molecules, and different targets even. I think we should probably start thinking about, you know, more of the customization, right? MG is also known as the snowflake disease. Every patient is different. So how do we, I guess, you know, position the right therapy for the right patient at the right time, right? And it's something that I think we should start thinking about now, in anticipation, hopefully, of, you know, some of the work ongoing will be successful. So I think we must start thinking of individualizing therapy.
I would like to address another major challenge when it comes to doing research trials, and that is that every company wants to select the patient population for their trial where they have the best chance of showing that their product works. And that means that many patients who are in need are not enrolled in clinical trials. And a perfect example of this is the fact that we know the most about acetylcholine receptor antibody positive myasthenia gravis. And so all of our primary endpoints are based on acetylcholine receptor antibody positive myasthenia gravis. And there are many patients who don't fit into that group. And there are other patients who fit into that group but have comorbidities and have other issues and therefore don't qualify. So a real issue, I think, for someone like Dr. Wolf and for the investigator is the fact that they want to be able to offer treatment to all of their patients, and they're often having to select a very few who qualify for a trial.
Yeah, I'll pipe in real quickly, and I agree with all those points. One thing about recruitment was raised as a challenge, especially with the crowded space. I can tell you, compared to 15, 20 years ago, we're better at it. And so if you look at the older trials, we often failed in recruitment, and it did not end up with the number of patients that we had hoped for. And that really continued for quite a while. But several things happened. One is we built up clinical trial networks with more sites. We started systems, and this includes the industry-sponsored studies as well. They had more sites involved. Backup sites are part of the plan in case recruitment lags. So when you look at the more recent studies, for the most part, recruitment targets have been met, generally on time. So we've really gotten better at that. And another thing that has changed also is the allowance for current therapies to continue through a trial. If you look at all the FcRn trials, or you even look at, say, some of the trials with the C5 cleavage agents, the pre-existing therapies were allowed to continue. And so that was true also with the NeuroNext trial, which involved the Clinical Trials Network sponsored by NIH. So we've gotten better, but it still can improve. The other thing I just wanted to mention real quick, industry is a crucial partner for clinical trials in MG, but not every study that has a therapeutic angle to it is something that industry is interested in. And so funding does remain a challenge. And, you know, I was part of a very, actually the largest, from the NIH perspective, funded trial for MG, which was the thymectomy trial. We're looking now at another trial that industry is not going to be interested in. And yes, we have concerns about where we're going to get funding for that trial. So that's another challenge.
Yeah,
go ahead.
I would like to add just one more point. I think also as more drugs are becoming available with endpoints or that are really targeted toward what is established as manifestations of myasthenia gravis, it is time now to listen more to the patient's needs. And they may see the disease differently from the way physicians see it. And I think that opens possibilities for designing new trials that are more focused on patient experience and what they suffer from.
So my question is how is the seronegative patient going to be included in future studies? Because I know we're a small margin, but we're left outside that box.
Yeah, I think there are more can and provide partial answer to that. I mean, Momenta is running trials where we don't exclude seronegative patients. I think what remains to be determined, because much less is known about these patients, is what is the mechanism of the disease? Is it that we are not looking for the right autoantibodies, or are autoantibodies playing a lesser role than in anti-AChR positive patients? So I think there is both a research angle to figure out what is the mechanism of the disease in patients who are seronegative, and then what are the approaches that you want to take with this patient that correspond to the mechanism of the disease.
I would like to also comment that there's an additional challenge with seronegative myasthenia gravis patients, and you yourself highlighted in that you went so many years before you were diagnosed, and that is the diagnostic uncertainty regarding seronegatives. Argenx also includes seronegatives in their MG trials, but again, there is less diagnostic certainty even among the principal investigators when they're discussing the seronegatives, because of the fact that that antibody that is sort of a wonderful affirmation that you are dealing with a problem at the neuromuscular junction is not present. And so you do other ways of trying to say that the patient is diagnostically still belonging in this category, but that does remain a challenge.
Well, part of the challenge from my perspective, and obviously doing research, is that so many patients are diagnosed with conversion disorder or some functional disorder prior to getting an accurate diagnosis. Through my research, I have found that some patients, like maybe 10, 12 years down the road, all of a sudden labs become positive, EMGs become positive, cause I don't know. But yeah, I just wish it was kind of more of a better diagnostic criteria at the earlier side, so treatment could be given before we go 10, 20, so many years, and we would get progressively worse.
An insult gets added to injury, and you're told that it's a conversion disorder.
Correct. Because now
And historically,
Mhm.
many diseases in neurology have gone that route until we've understood them better, so that we can actually validate patients' complaints.
Thank you.
I just
Go ahead, but I just want to mention there's
yeah.
quite a few comments and questions. I don't know if you guys have your Q&A and chat boxes open. We need to address those. Go ahead.
Right. Yeah. So it looks like the first one that came up earlier was for you, Dr. Wolf, about the FcRn receptor inhibitors. So the question is, can you discuss how this class of drugs may impact the lives of individuals with MG? Do you anticipate that MG treatment paradigms will change as a result of these therapies?
So the first point I'll make about that is there is a potential for wide applicability of these agents across a variety of different IgG-mediated disorders, and that includes several different subpopulations of myasthenia gravis. Even seronegatives, and one point about seronegatives, a lot of the seronegatives actually have antibodies. It's just that our conventional techniques for detecting them miss it, okay? And I'll show a little bit of data on that when I give my talk, all right? So in addition to that, where would we use these agents? Some of them are IV, some of them are sub-Q infusion, but where could we use these? So we've been talking about bridging therapy some. Really, that's a term I didn't even hear until 6 months ago, 6 or 12 months ago. We have drugs that work in a large majority of populations of MG, but they can take a long time to really start having an impact. They don't work in everybody. Nothing works in everybody. But these agents, these FcRn agents, which work quickly, they seem to be quite safe so far. They have broad applicability across different MG populations. It could be something that does change that paradigm. And look, steroids work in MG, but they are hard to take long-term, especially at high doses. And we have a paper that just came out showing that the impacts of prednisone as far as adverse events, even at lower doses that we were pretty comfortable with, it's not a clean slate. They do cause problems. That was an outgrowth of some of the thymectomy data. So, occasionally there's a pro-con in the literature. Use steroids in MG, don't use steroids in MG. Most people who take care of MG patients and most MG patients will have ended up on steroids at some point. But could we shrink that? Could we really reduce the use of these agents that, while they do work, have a high burden of side effects? I see FcRns maybe replacing that transition there in that bridging moment. And also, more than just a moment, that bridging era of time. The other thing is, for crisis situations where our standards of therapy don't work so well, or IVIG is not available, or plasma exchange is just not done, maybe they can work there, because you will see improvements quite quickly with those agents. I'll stop there. There were a couple other quicker points. One very quickly, though, that others should discuss. Why is there this increased excitement for MG? And part of that is it's a pretty well-defined disease. We understand it reasonably well compared to other immune-mediated disorders. So industry is going to have an interest in that. Anyway, I'll stop.
Thank you, Dr. Wolf. I think we touched upon the next question, which is why do most trials not include seronegative patients? I think multiple people on the panel have addressed it. It's because it's tough to have an endpoint when you don't have a beginning point or a defined number or objective data, which we're trying to get around by finding these antibodies that these people are positive to and trying to come up with the panels. So another related question is, why don't we have an MG panel that includes all the new antibodies that lupus has? Is because we haven't discovered all of these antibodies yet. personally in practice, I think I will try to include Agrin more often now. But it doesn't actually change my treatment currently because I'll still treat them with the immune therapies. I don't know the experience of the other physicians on the panel with the seronegative patients and looking for these antibodies, or making your own panels actually with your miscellaneous labs.
I think a couple of points maybe I want to mention here is around seronegative patients, and yes, we may know of other antibodies involved, but it has to do with the pathology and the proposed mechanism of action of the agent that we're using in a clinical study. So it has to be justified. There's a benefit risk, proposal that we have to make, and has to be accepted by regulators and ethics committee and all of that. And of course, we're trying to optimize the study and the patient population through criteria that increases the chance of seeing a signal. And that may not be, some of us may look at that and say, well, this may not be ideal for all the patients. But truly it's just the nature of drug development. You start with a selected, patient set and hoping that you'll see a signal. And then, of course, over time, you start, loosening the criteria and expanding to a broader patient population. I think the example from Dr. Wolf earlier on, how we're now, including patients and they can stay under background therapy and so on and so forth. So I think, hopefully the community will start expanding and addressing a broader patient population. But some of it is just the nature of drug development.
I completely agree with those points, and I just want to add that this idea of searching for signals, sometimes that comes from more partnership externally. So I see that there's a question about investigator-initiated studies, research that is involving real-world evidence or patient registries. These are all ways where we can potentially find signals that then would support a more formal drug development process and clinical trials for seronegative patients.
Also, with regard to the question of why more potential antibodies aren't studied widely, a lot of that has to do with the fact that if a test has not been fully validated, you may have a suspicion, for instance, that Agrin antibody is actually pathologic in the progression of myasthenia gravis. But until you know at what titer, at how the test is being performed, how reliable the test is, you can't really make decisions based on it. And so there are many investigators, and I think that's part of the role of investigator-initiated studies, that are actually trying to further develop tests that can validate and identify new antibodies that are present in the seronegative population.
Okay, so on that note, I think there's one more question here about, there was a patient here who was on a phase 3 trial of FIRDAPSE earlier this year. There wasn't a mention of COVID-19, though it was gaining speed. Why is, I guess, the results of the study or the study on FIRDAPSE not more widely known? I don't know if anyone knows more about the FIRDAPSE studies going on right now.
I heard that it was on hold, so, but I couldn't give more information than that. I just read that on the internet.
Yeah, same. Okay, so a question for everyone on the panel, and I think to echo some of the things going on in chat about being optimistic about treatments in MG. What are the exciting therapies that are on the horizon for myasthenia gravis from each of your perspectives? It's a big question, yeah.
I think it's more appropriate if someone not from pharma answers that. So,
Yeah.
yourself or Dr. Wolf?
Yeah, I mean, for me, having the ability to use the complement inhibitors in pediatrics is we're right on the cusp of it. for using eculizumab, especially for the refractory patients, and then the other like antibody like specific monoclonal antibody directed things other than just IVIG. I think that's exciting. And then having treatments from all the other different mechanisms as kind of previously discussed, that's really exciting to like learn more about.
Yeah, there's going to be a couple of talks later today that I hope everybody can stay on for, that are going to discuss clinical trials.
Oh, yes.
The very next session is going to be a discussion of the clinical trials. A few things that I have a couple of slides. If there's time, I'll show, but potential uses of IL-6 antagonist drugs, which are actually coming into play a little bit with the pandemic.
Right.
Could they potentially play a role in helping MG? But there's a lot of activity already, as you'll see. Yeah. We mentioned FIRDAPS, diaminopyridine, and you know, the Musk data for that agent looked pretty good, and so there's excitement on that level as well for that population of MG patients.
What I saw mentioned on the comments about fatigue in myasthenia gravis, and how people sometimes perceive patients as being lazy. I think it's a very interesting discussion of fatigue and neuromuscular disease in general, and how elusive it is, and how much we would all love to be able to study it in more detail and study it in a way that we can demonstrate something that makes a difference.
I think.
All right. Yeah.
I'm sorry.
Oh, sorry. Go ahead. Go ahead.
I guess from my perspective, and probably many others, is finding physicians that are knowledgeable in what antibodies to test and what pharmaceuticals are available to treat MG. Because like I live in South Florida, and the local neurologist is just a local neurologist. They don't have that MG specialty title to really know. I have to go outside the area or back to New York to really find an expert.
And
that's actually, yeah.
Internet, with the internet and with connections that are now existing between true experts like are on the panel here and community neurologists, there is better communication now. And I see many of these blogs where a community neurologist will say, I have a very challenging patient. I read one just this morning, as a matter of fact. I have a challenging patient who has rheumatoid arthritis and myasthenia and has failed multiple agents. And now he put that question out in the blog, and already advice is coming in from key experts. And I think that one of the beauties of being connected in this internet social world is that it will be possible for patients to find good treaters, or at least treaters who are willing to learn from experts.
Yeah, I guess part of the problem is so many patients cannot travel due to either like the fatigability or financial aspects that's associated with the MG. So if physicians could do telehealth and consult with the local physicians and read files, that would be so helpful.
And Francis, I think it's a good point. It's an ever-present challenge for those of us who design clinical trials to think about what is the burden of the study and the visits on the patient population, and does that actually exacerbate aspects of their condition or disease? So I do think that, with my fellow panelists, we are committed to trying to make trials more efficient, reduce burden on patients, look for either technology or alternative designs that try to simplify the experience as much as possible, but still provide the necessary structure that we need to have to conduct good research.
And actually, like a question that patients and families may be interested in is, how can they get more involved in clinical research themselves or help accelerate the drug development process for MG?
Yeah, this is a really great question, and I believe the patient community is already doing a lot with some very active patient association like this association or MGFA. And you know, as Frances was saying, I think is really questioning neurologists and pushing to get answers, that is really key to making progress. What I think a lot of pharma also are doing now is listening very closely to what patients have to say in terms of what is the need of the patients, how can we address these needs with drugs, and that should result in significant progress in the next few years.
I agree. I think patient centricity is a key, and from a clinical development point of view, we need to involve patients and their families from very early on. And we've seen, that trend, we've been observing that in the past few years, where the opinion and the feedback from patients and their family is being integrated from very early on, even the study design or even at the concept sheet, and trying to understand their experience on a clinical study. What does it look like? And you'd be surprised. I mean, we've heard and seen some cases where the feedback from the patients and their families actually altered the study design. I just think we need to do more of that and continue to listen.
Yeah, and
in terms of what a patient's family can do to gain access to clinical trials, you have to advocate. Patients and families and patient physicians have to advocate for their patients to find out where is the closest clinical trial, what clinical trial is conducive to the convenient enough to a patient, manageable enough in terms of distance, and might conceivably help that patient for someone who really is motivated to find something additional.
I think another aspect of another aspect that is recent that patient can be involved with is that, for example, the FDA and also the equivalent in Europe wants to hear from patients when they review drugs for approval. And this has been in place for a few years now, but it is making a big impact in terms of taking into account the voice of the patients for drug approval and as a consequence also for drug design.
And the FDA is now more open to early access programs, so that if a compound has been tested to a degree that safety is felt to not be an issue, the FDA will often allow, if a company is in agreement to allow it, early access before something is fully approved for use. So that's something to also ask about and inquire about.
Lastly, I would just say that most of our companies, on our websites, we also have the opportunity to input questions or connect directly with either our medical group or clinical group if you are interested in participating in a patient panel or something of that nature. So I would definitely encourage using opportunities like this to learn about the companies that are doing research in this area, and then exploring their websites and potentially putting your name out there if you'd like to participate.
I've been typing in the question.
Yeah, there was a specific question for you again. Yeah, go ahead.
Let me just say something also about seronegatives. The trial I mentioned before that we're looking at, it would be an ocular MG, and many ocular MG patients at a higher proportion than generalized MG patients are seronegative. It's 40%, 50% in some series. So excluding seronegatives in a trial of ocular MG, you're really going to restrict your population. So we have plans in place to really try to include those seronegative patients. And I think there are ways to ensure investigators and authorities, regulatory authorities, that we really are dealing with immune-mediated myasthenia gravis and not something else. One thing that comes up, if you're looking for patients early in the disease course, As time goes on, we can become more confident with the diagnosis. That's not just true in MG. That's true in a lot of areas of neurology. Initially, it can be pretty hard for us sometimes in complicated patients to know exactly what we're dealing with. But over time, it becomes more clear. That's true in neurodegenerative diseases. It's true in a lot of other areas of neurology as well. And it falls into myasthenia gravis and the question of congenital myasthenic syndrome and other things. But anyway, they're even early in the disease state, we're coming up with some strategies to include seronegatives and be confident we're dealing with an immune-mediated disorder. All right, so I'll leave it at that.
Okay, great, thank you. So just to kind of wrap up, because we're kind of hitting up on the time. To get involved with clinical trials, just like the people on the panel said, you can go specifically to the websites of the trials you're interested in through the pharmaceutical websites or through clinicaltrials.gov, or by connecting with your MG specialist or finding an MG specialist, because we are aware of the clinical trials and maybe running our own clinical trials as a site. And so MDA has that information, as Nicole went over earlier, and as well as the Myasthenia Gravis Foundation. As far as like specific clinical trial criteria, that depends. So we'll kind of leave that for an offline discussion, but thank you all on the panel for having such a, very productive discussion. I think we got to most of the patient questions, but Dr. Wolf is actually going to be going over a lot of the questions that came up during this panel in more detail along with slides. So thank you all for a very productive discussion. And I think Nicole, you're going to kind of take over from here. Is that right?
Yes, thank you, everyone.
Okay.
Thank you very much. We are going to take a quick break and be back at the top of the hour. So thank you all very much. It was a great panel discussion.
Thank you.
Thank you.
Thank you very much.
Thank you, everyone.